Hello, good morning, all of you. This is the welcome to Lakshya Academy, and this is the part 14 of excretory products and their elimination. Okay, in the previous part, we learned what is GFR and what are the different types of renal substances, A threshold substances, high threshold substances, low threshold substances. We can classify the substances present in the renal filtrate or tubular fluid into three types. If they are highly helpful to our body, useful for our body, they are called high threshold. If they are not at all useful, they have to be eliminated. They are called A threshold substances, and some substances are little bit they have to be taken into our body. They are low threshold substances like urea, uric acid are low threshold. A threshold are creatinine. This is the next continuation part. The first part you learned in the urine formation is ultrafiltration or glomerular filtration. The second step is selective reabsorption and secretion. Simultaneously occur these two steps: selective reabsorption and selective secretion. Reabsorption and secretion, these two occur simultaneously. About 85% of the filtrate formed is reabsorbed in a constant and unregulated fashion, obligatory or mandatory by PCT and descending limb of Henle's loop. And the reabsorption of the rest is regulated by that is called obligatory. The reabsorption of the rest of the fluid is regulated. That is not obligatory. This is obligatory. Next, tubular cells. This is one of the 2 marks question. This one, tubular cells secretes substances such as H plus, K plus, ammonia into filtrate. Tubular secretion is also an important step in the formation of urine because it helps in maintenance of ionic and acid base balance of the body fluids. Look at this one. 85% of the filtrate formed is reabsorbed. I will show you here. This is a glomerulus. The filtrate is formed here in the lumen of the Bowman's capsule. This is the lumen of the Bowman's capsule. From the as the filtrate passing down here, some of the useful substances are reabsorbed. Here, how much reabsorption? About 85% of the filtrate is reabsorbed. In the digestion lesson, you learned absorption. Here it is reabsorption. How much per filtrate? 85% of the filtrate formed is reabsorbed in a constant, unregulated. Here, no regulation of hormones, unregulated fashion. And this kind of reabsorption takes place in the PCT as well as the descending limb. PCT as well as the descending limb. The reabsorption that takes place in these two parts is called as a obligatory reabsorption or mandatory reabsorption. Where does it occur? It occurs in PCT and descending limb of Henle's loop. The reabsorption of rest of the fluid is regulated. It is unregulated. That is regulated. Next, tubular cells. Tubular cells means, what does it mean? The cells present in this renal tubule are called tubular cells. What is the type of epithelium here? Can you remember that? Here simple cuboidal epithelium with brush border. Here what type of epithelium? Simple cuboidal epithelium without brush border. What type of epithelium here? Simple cuboidal epithelium without brush border. What type of epithelium is present here? Simple squamous epithelium. Here simple squamous epithelium. Here simple cuboidal epithelium. You have to know this. Nephron means up to only here. This part is not a nephron. This is the collecting duct. Collecting duct is not a part of nephron. Tubular cells, that means the cells present in this part secrete. One important thing you have to understand here, I told you in the previous video also, If the substance is coming out of this tube, that is called as, if something is coming out, okay, that is called as, what do we call that? That is called reabsorption. If something is entering into this one, this tube, yellow color one, okay, that is called secretion. Okay, secretion is always active process. Here something is secreted, here some substances is secreted, here some substances are secreted, but it is not shown here. Okay, if something is secreted here, okay, that is called, okay, that is an active process. That means it utilizes ATP. If something is coming out from here, this is called reabsorption. Okay, you have to understand that the inward arrow, this is this arrow indicates secretion, this arrow indicates, these all arrows which are coming out from this one, this is it indicates a reabsorption. Okay, the tubular cell secrete substances like H plus, K plus, NH3 into filtrate. Observe here. The tubular cell secrete H plus, NH3 plus, K plus into the filtrate. In this tube, what is present? Filtrate is formed, not urine. Primary urine is present. And the tubular secretion is an important step in the formation of urine because it helps in maintenance of ionic and acid base balance of the body fluids. Our body fluids acid base has to be maintained properly. Okay, you learned earlier what is the pH value of our blood. Our pH value of our blood is 7.4. Any disturbance in the, okay, this as a pH value can lead to death of that person. Okay, right. Next part. In the proximal convoluted tubule, the breast border cuboidal epithelium of PCT increases the surface area of reabsorption. Nearly all the essential nutrients you can see here, all the essential nutrients, and 70 to 80% of the electrolytes and water, Na plus, Cl minus, K plus, these are electrolytes and water are reabsorbed in the PCT. Nearly all the nutrients, glucose, amino acids, vitamins, how much electrolytes? 70 to 80% of electrolytes and water are reabsorbed where in the PCT. How they are absorbed? This was given in previous NEET exam. Okay, reabsorb Na plus. I told you earlier, reabsorb means the arrow coming out. Na plus is absorbed by which process? Active process. Cl minus by passive process. Glucose and amino acids by active process. Water by osmosis. The water reabsorption that occurs here is obligatory reabsorption. PCT also helps in to maintain the pH and ionic balance of the body fluids by selective secretion of H plus and ammonia. You can observe here, this is H plus and ammonia are secreted into the filtrate. They are secreted into the filtrate, and by the reabsorption of HCO3 from it. What is reabsorbed here? HCO3 minus. Okay, anion. Okay, water secreted here. H plus cation and NH plus cation are reabsorbed here, and these two are secreted here, and these all are reabsorbed from this PCT. Okay, now next part, PCT completed. The next part is in the loop of Henle or Henle's loop. Okay, Henle's loop. It was asked in previous NEET exam 2 to 3 times. The reabsorption in this segment is minimum. However, this loop of Henle is very important topic. What would happen if loop of Henle is not present? That kind of application questions were asked in previous NEET. If there is no loop of Henle in our nephron, the person produces a dilute urine. Generally, our urine is concentrated. Our normal body fluid osmolarity is, what is meant by osmolarity? The amount of solute present in our body fluid, that is called osmolarity. For example, if you take 100 ml of water, you put some salt or sugar, that means that water has some osmolar, the amount of solute, that means that sugar is a solute or salt is a solute, how much that solute is present in that fluid, that is called osmolarity of that fluid. Okay, this Henley's loop plays an important role in producing our urine osmolarity 1200 milliosmoles. Our urine osmolarity 1200 milliosmoles. Our body blood osmolarity 300 milliosmoles. That means our urine is 4 times concentrated than our blood. 4 times. Okay, reabsorption in the loop of Henle is very minimum. However, this region plays an important role in the maintenance of high osmolarity of the medullary interstitial fluid. Medullary interstitial fluid. Okay, what is the meant by medulla? This part is called medulla. This is the inner part of the kidney. This part is medulla. This is the outer part of the medulla, and these all parts are present in the cortex of the kidney. This is the inner outer part of the kidney. This is the inner part of the kidney. Here the osmolarity. Okay, the osmolarity you can observe here. Here the osmolarity 1200 milliosmoles. Here 1200 milliosmol. Who is playing an important role in this maintaining high osmolarity 1200? Henley's loop plays an important role in maintaining high osmolarity. Okay, Henley's loop this plays a significant role in maintaining high osmolarity of the medullary interstitial fluid. And osmolarity is the measure of, what is meant by osmolarity? The measure of solute concentration, defined as the number of osmoles of solute per liter of solution. In 1 liter of solution, how much, okay, how many number of osmoles is present, that is called osmolarity of that fluid. This is the symbol. Okay, these are the OSMOL, means osmolarity for liter, or you can write OSML, osmolarity for liter. The descending limb of Henley's loop. Okay, I will complete the remaining part in the next video. uh, okay, you learned here reabsorption in the PCT, selective reabsorption and selective secretion. About 85% of the filtrate is reabsorbed in a constant, that is called as a obligatory or mandatory reabsorption. Tubular cells secrete okay, different types of substances like dissolve. And what reabsorption in PCT, brush border PCT is made up of what type of cells? Brush border cuboidal cells. And where is PCT? PCT in the cortex part of the nephron. Nearly all the essential nutrients, 70 to 80% of the electrolytes and water are reabsorbed in the PCT. Na plus is reabsorbed in active trans by using ATP. Cl minus without using ATP passive. Glucose amino acids reabsorption occurs in active manner. And water, very important one. Osmosis, it was asked in previous NEET exam. Na plus. And PCT also helps in maintain the pH and ionic balance of the body fluids by selective secretion of H plus and ammonia into the filtrate, and by the absorption of HCO3 minus from it.